now focusing on the adaptive immune system. So chapter 17, so for adaptive immunity, we're actually going to discuss a bunch of different topics, which are covered throughout chapters 17, 18, and 19. I'm not going over in full depth for those 3 chapters. And in fact, I'm not even going to talk about any specific immune disorders. Allergies are going to be super brief, so just barely touching on some of the topics. But I wanted us to spend a good amount of time talking about vaccines. And then you get a little bit of the blood system, but most of that will be covered in your lab. You'll be working with that in lab. So make sure that you pay very close attention to the adaptive immunity lab intro, because there are parts that I elaborate more in that than I will in this video. And so I've kept in mind what I've already covered in the lab, and so I expect you to watch both of the videos closely, carefully, take notes. Okay, so I tried to let you know where you could look in your textbook if you wanted to see images, read the text, to get more ideas, and also then at the end of the chapter see some practice questions, right? So we'll start with chapter 17, and then it'll kind of get mixed up a bit. But this is how I think the topics actually relate to each other and flow the best. Okay. So adaptive immunity, as you recall, means that you have stimulated the immune system to respond specifically to the exact pathogen that is triggering that immune response. And this is carried out by the cells called lymphocytes. And remember, because we have a large amount of these cells that reside in the lymph nodes, that's why we call them lymphocytes. These are the T and B cells. These are the T and B cells that we'll be talking about. I know with the innate immune system, we talked about a lot of different components of the innate immune system. For the adaptive immune system, you can basically just break it up into 2 branches. The first branch being what we call humoral. This name comes from old medical practice when they believed that a person's health was the source of health were all of the fluids of the body. They would do what we would find to be very absurd and strange practices, like draining blood, or different things to do with fluids, as they were trying to figure out how the body worked. Well, this term humor for kind of started to be known as like health, and so then as scientists learned about antibodies, and that we could detect some antibodies in the blood and in the lymph, they decided to name this branch humoral branch, because it does relate to some of the fluids of the body, right? So you can see how that's related. Okay, so the humoral branch that's responsible for these producing these antibodies is we call it the B cells. All the B cells will have receptors, which are essentially antibodies bound to the surface of the B cell. And then when that comes in contact with the exact antigen that fits, remember that antibodies and antigens fit together like a lock and a key. And so when the antigen lands on the B cell that is a match, this will cause the production of lots of antibodies. Well, that's a very simplified view of it. We'll talk a little bit more about that in a little bit. And this process typically takes 7 to 10 days for the B cells to be able to really make those antibodies. And we'll see why that is in a little bit. Okay, so now keep in mind, we talk about the adaptive immune system, we talk about the two branches, but everything's always going to be interrelated. Everything's going to work together, right? Our body is it's one body with all the parts working together. Okay. The other branch of the adaptive immune system is called cell-mediated immunity, and this is attributed to T cells. And we'll see that there are different types of T cells, and they all play an important role, but we tend to, if we had to really boil it down to one thing, we would say the T cells that specifically kill infected or cancerous cells, although there are more functions than just that, because we have more types of T cells than just this one type. And so again, remember that both of the branches they're part of the adaptive immune system, so therefore they're capable of providing lifelong immunity because it's specific, and there typically will be a memory response that gives you that specific protection long term. Okay, the innate immune system, on the other hand, is not specific, and it is not going to protect you in a specialized way. You can keep having the same pathogen over and over again, and the innate immune system will respond exactly the same way each time because it doesn't know that it's the same pathogen over and over again. Whereas the adaptive immune system has these memory cells and will immediately respond to where it'll prevent the infection, you won't even have symptoms, etc. So very different overall. But again, they still have all the different parts do end up, interrelating and helping each other out in one way or another. Okay, so this specific or adaptive immunity takes time, right? As you're exposed to the antigen, whether it be being exposed to the regular pathogen, getting sick, or whether it's getting exposed just to the antigens, such as through vaccine, each time you encounter an antigen, your immune system gets to respond to it. Both T and B cells will have actions and provide you this long-term specific immunity. So it takes time, right? Because you're not going to be suddenly introduced to all the antigens all at once, right? So it's built over time. So often the adaptive and innate overlap each other, as I've been saying. So inflammation comes up in all of our lectures, right? The complement system as well. So you'll see that as a commonality for both types of immunity. As the cells of the adaptive immune system are working, the innate systems are also working, right? And in the last lecture, I think I told you can think of it as the innate is like they're like the army. They're on the ground fighting physical, right? You can think of it, if you think of it as like a war, those are the troops that are on the ground doing the hand-to-hand combat. Whereas the adaptive immune system will be like your air support and your, I don't know a whole lot about wars really to use this as a great example, but things like bombs and air support and things like that could be the adaptive immune system. It's not going to be necessarily immediate. It might be, right, if you already have the immunity, but the adaptive immune system is like the big powerful part of our immune system. The innate is always going to be functioning, and in a sense is buying you time for your adaptive immune system to ramp up and do its part. Okay, so that's the dual nature of the adaptive immune system, right? So both T cells and B cells arise from bone marrow, just like all of the cells of the blood that we talked about, right? The B cells stay in the bone, and that's why they're called B cells. They mature. You make sure that they're not going to attack your own cells before you release them, and then they'll go to the lymphatic system, right? T cells, on the other hand, go to the thymus and mature, and that's where you make sure before you release them that they're safe. You won't have autoimmunity, right? And then once those are mature, then they will travel to the lymph tissue, right? So then both the B and T cells will reunite to do their job. You will have a lot of them in lymph nodes throughout the lymph tissues. And then also remember the spleen is another major reservoir, but the spleen is technically part of the cardiovascular system. And so you'll have some of these circulating in the blood as well, and throughout your tissues, but the vast majority, the biggest concentration will be in the lymph tissue. Okay. So a little bit more about B cells. So we just said they begin in the bone marrow, they stay in the bone marrow, they mature in the bone marrow. When they are mature, they have their receptors, and we've checked to make sure that those receptors will only respond to foreign antigen, will not respond to any of the cells of our body to prevent autoimmunity, right? It'll then travel to the lymph. Some of it will be in the blood system, but most of it will be in the lymph, right? Also recall that the big clusters of lymph nodes are the tonsils and the Peyer's patches in the gut. Okay, detect antigen. So if antigen is detected, if one of the B cells finds its perfect match, it's going to become activated. It's going to take in that antigen, process it, and display it. And then there's some other parts that have to happen, but eventually that activated B cell will make a whole bunch of copies of itself. We call that clonal expansion. And then some of those clones will become plasma cells, which are just antibody factories. They just make and spit out lots and lots of antibodies. And then another part of that army, that clone, will become what we call memory cells. And they're going to hang around throughout your lymph and throughout your body, so that if that antigen gets access to your body again, it'll immediately be able to secrete large amounts of antibody and give you that secondary and lifelong protection. Okay, so there's a little bit more detail to being able to get to that clonal expansion part, right? And this is like a check and balance to make sure. So we've had the first check and balance during maturity, that our B cells will not react to our self and hurt us. But then we also have this another kind of check and balance, and that's with using the helper T cell. The helper T cell has to basically give permission to the B cell that has encountered its perfect match. And so what happens is the B cell, maybe it's in the lymph node, and an antigen comes by and sticks to it. And here the antigen is still attached to the bacteria, but it doesn't even have to be the whole pathogen, right? It could be just the antigen or a piece of bacteria, right? So any form of antigen will do. But it has to be the specific antigen to bind to this receptor. You have billions of different B cells. Each B cell has its own unique top portion of the receptor that will only fit with one specific type of antigen. And that's why it's that like lock and key fit. Okay. So this is a very special event. This antigen could have passed hundreds of thousands of B cells before reaching the correct one that binds. Okay. So now we have this B cell that says, wow, finally my time has come. I've found my match, right? And it's going to then, we call antigen process and display. And the display of this antigen is going to be presented on the MHC2 tag, right? So remember, all the white blood cells, immune cells, have MHC class 2 receptors, which are the self tag, saying that I'm an immune cell, okay? And then it displays the antigen from the pathogen, just a part of it, on that tag, saying, hey, I'm an immune cell and I have detected an antigen. Please give me permission to do my job. And this T cell, who has also seen this antigen, has this helper T cell has already been activated, we say. And it's saying, yeah, this is an actual true problem. You're not doing something wrong. You're correct. You detected this antigen and we need to do something about it. And so the helper T cell releases cytokines. And those cytokines tell the B cell, yes, go ahead and make a clonal army, right? Clone yourself hundreds and hundreds of times. And each of those clones have this exact receptor for that exact antigen. And most of those cells will be, this is switched around. Most of the cells will be plasma cells, which secrete the antibody into the lymph and the blood right throughout the body to bind to any antigen that's anywhere in the body. And then these memory cells will have the bound antigen, the receptor form, and those will go throughout the body to provide lifelong immunity in case that antigen ever shows up again. These are ready to just immediately turn into plasma cells. And then you'll also get more memory cells. And so each time you'll have an even stronger and stronger and faster reaction. Okay. So in order for the B cell to undergo clonal expansion, it has to have the helper T cell has to release cytokines in order for it to be able to do this part. Okay. And so I wrote that over here. All right. Now, there are situations where a B cell can secrete some antibodies without that full process. And this is only in the case of large repeating units of antigen, which are typically polysaccharides, such as LPS. And it basically binds to several of the receptors on the B cell, and the B cell says, whoa, we have a problem. I just got, not only did I find my perfect match, but I found it over and over and over and over again, like at the same time. We must have a big problem. Like we must have a big infection. I'm getting so much antigen. And so it actually turns on this other pathway to be able to immediately secrete some antibodies that will be a perfect match to this. But this is a relatively small amount of antibodies. And it will clone itself a few times, but again, it'll be a relatively small amount compared to the full-blown reaction that we could get if we have a helper T cell. So without a helper T cell, and only in the presence of large repeating units of antigen where you get multiple binding to the receptors at once, you can immediately get a small subset of plasma cells to secrete antibodies. You will not have any memory, okay? So no memory cells are produced, but you get this immediate reaction, and that's it for this. Okay, so antigens and antigen and antibody binding in the body, you have agglutination, which we actually kind of see here if we had, if there were more of these polysaccharides around, they could bind to the rest of this. And so this one cell would bind, multiple pieces of that antigen. And so whenever you clump something together, you get a bunch of the antigens and antibodies in one spot. That's agglutination, clumping, okay? Neutralization is similar, but now the idea is that you have a bunch of small, either toxins or viruses, and you want antibodies to bind to those and take up the receptors so that the antibody is preventing that either the virus or the toxin from being able to get into a cell and cause damage, right? So that's neutralization. And then eventually it will get taken up by phagocytes. Opsonization is when you coat the surface of like a bacteria, for example, you coat it in antibody so that phagocytes will be able to come and eat it up more readily. Okay. And we already learned how antigens, if antibodies bind to it side by side, you can have the classical pathway of complement activation. So activation of the complement system can occur due to antigen-antibody binding. It's not always, right? It has to meet those conditions of two antibodies side by side, and then the complement protein in the blood comes along and binds, right? And then you'll get the activation, the classical activation. Okay, so we had talked about all of those before. What we have not talked about before is antibody-dependent cell-mediated cytotoxicity. And this is where I cannot perfectly put this into any of the groups, right? Because now it's antibody-dependent. So we're using the humoral branch for antibodies, but it's cell-mediated, which is the other branch of adaptive immune system, right? And then we'll see that we'll actually get a bunch of cells that respond, including some innate cells. So it's really it's involving all the parts of our immune system. And the reason for this is that you have a parasite. Typically, this would be a helminth, like a helminth worm, not like a earthworm that you see outside, much smaller than that, but much bigger than our cells, right? Here's a macrophage, which is a large cell for our body. And here is this parasite. It's way too big to be eaten up by this macrophage, right? And so in the case of these large parasites, or which the correct term is extracellular, right? Because it's outside of the cell. And antibodies start to bind, and then that's going to trigger macrophage will come and see this antibody bound, and it's going to release some of its toxic substances. The eosinophils will also be able to bind to those antibodies and release its toxic substances and enzymes. So eosinophils specifically release the lytic, right? We talked about things like protease, lipase, right? All those different enzymes for digestion. When we talked about, like in the macrophage, for example, when we talked about phagocytosis and how they can digest microbes. So all those things will be released. So we have cytotoxic cytokines, are all these blue guys. So we're releasing cytokines to get a bigger response, to get more of these cells, recruit more white blood cells to fight. And then they're also releasing their lytic enzymes and their porphyrin, right? Porphyrin pokes holes, starts to make damage, and helps the other enzymes to be able to go inside and do their job. And so we see eosinophils, we're going to see natural killer cells. And so these are innate immune cells that are coming to battle, but they only know to do this because of the antibodies that are coating the parasite. Okay, so this is why it's antibody dependent, right? Antibody dependent cell mediated cells of the innate immune system, cytotoxicity, releasing toxic substances from the cells to damage this parasite and kill it. Okay, so here's the summary of all of those outcomes of antigen antibody binding. So I wrote the list here. Then we talked about what antibody dependent cell mediated cytotoxicity is. And now this is the image to summarize once again all of the possible outcomes of antigen antibody binding. So you can have agglutination where you clump together, right? In this picture, they're clumping together bacteria. It could be viruses. It could be large pieces of antigen, right? Anything that the antibody binds to can agglutinate. We can activate the complement through the classical pathway that we talked about. And eventually, that can lead to formation of the MAC complex, right? Membrane attack complex, which will lead to lysis bursting open, right? The bacteria. We can have opsonization where we have coated the bacteria and made it much easier for the phagocyte to grab onto and eat. We can have the antibody-dependent cell-mediated cytotoxicity if it's a parasite, extracellular parasite that we're dealing with, right? And the neutralization, which we typically think of neutralization as a virus or a toxin. You technically can do it for a bacterium too. What neutralization requires is that all of the receptors get bound to antibody. So now this virus, it is now inert. It cannot infect a cell. It doesn't have anything to bind to a cell anymore. Same thing with this bacterium. This would mostly be like intracellular bacteria where you would see something like this. And then toxins as well, you want to have those all bound so that they cannot cause any outcome. Okay, so neutralization is making things inert, and yes, eventually phagocytosis will occur. But opsonization, you don't necessarily get coating of the entire pathogen. It could be just one or two antibodies, and it's still going to help phagocytosis, right? Okay, so hopefully I've clarified that for you. Each of these figures are labeled, so you could look up and read more about it if you need to study up on these. Okay. All right, so now we finished our discussion on B cells. We will come back to that when we talk about vaccines, in the sense that we'll be talking about making antibodies, but we're pretty much done thinking about the B cells in particular. And now we're going to focus on T cells, cell-mediated immunity. So antibodies are super great, as we just saw in this previous image, right? They help us do a lot of different things to get rid of infections, but they cannot, right, directly take care of intracellular pathogens, right? Antibodies are not going to get inside of our cells. So if you have an intracellular pathogen, it's already inside your cells and causing infection. The antibodies are not going to be able to get to those pathogens that are hiding inside of your cells. Once those pathogens come outside of the cell, the antibodies would have a very short amount of time to get to it before it gets inside another cell, right? So antibodies aren't very helpful when it comes to intracellular pathogens. Also, what about getting rid of abnormal cells of the body, cells that have started to become, tumorous or cancerous, or cells that have been infected that we want to get rid of so that the infection doesn't continue to spread? Antibodies are not going to help with that. Okay, so for these reasons, this is why we need the cell-mediated immunity. And for the most part, we'll be thinking of these cytotoxic T cells that come and kill infected and weird cells, right? Each T cell is specific for a certain antigen. So it is still part of the adaptive immune system. It is specific. Once mature, the T cells leave the thymus and reside in the lymph tissue. Remember these properties. Okay. So the overall purpose of T lymphocytes is for the cell-mediated adaptive immunity. They mature in the thymus. They depend on the hormone thymopoietin to be able to mature in the thymus. They provide specific immunity, and they do not have anything to do with antibodies. They do not make antibodies. T cells also cannot interact with free antigen. They have to have antigen presented to them on a self-tag. So this could be a professional antigen presenter, right? An APC, which presents on the MHC class 2, or it could just be, random cells of the body that have been infected or have gotten weird, and they would be telling on their MHC class 1 tag, right? So cells of the body, MHC1, white blood cells, MHC2. All right. So T cells do not interact with free antigen. They have to have the antigen presented to them on an MHC tag. All right. So what are antigen-presenting cells? These are specialized cells that are able to digest and display antigen, right? So antigen fragments can then be presented on the self-tag, which would be the MHC2. So which cells are capable of doing this special antigen processing? B cells, which we already saw. Dendritic cells can do it too, and activated macrophages can also do it. And so then this really combines both innate and humoral immunity to activate the cell-mediated immunity, right? So T cells require an antigen-presenting cell to display antigen to it in order to activate it. Once activated, they can become effector cells to do the killing, or they can be memory cells for long-term protection. So one more time, T cells begin in the bone marrow, travel to the thymus, and mature. We have a checkpoint, make sure that they're not going to react with any of the cells of the body. Then we release them into the lymph, and they wait to encounter antigen. If they encounter their matching, if antigen is presented to them, they become activated, and then they could become a helper T cell, a killer T cell, which we call a cytotoxic T cell, a regulatory T cell, which makes sure our immune system doesn't go overboard and cause too much damage to ourselves. And then we also have the memory cells, right? So there's a lot of T cells. We're going to focus on 4 major groups, and we're going to be very broad with this, because you could take an entire course on the immune system. It's super complex. You learn about all the different specific types of cytokines and interactions of these different cells, but we're going to keep it as broad and as simple as possible. So regulatory T cells, we'll just say there's one type, even though there's several. Overall, the purpose of regulatory T cells is to prevent hyperstimulation. Remember, if the immune system becomes overstimulated, you can hurt yourself, you can even kill yourself with such a strong response, okay? The other problem is autoimmunity, where we don't want to start attacking our own, thinking that our own cells are pathogens, right? So hyperstimulation would be in response to a pathogen that is actually a pathogen causing the stimulation, But if your immune system responds too strongly, you get very, very sick and can even die. Autoimmunity, there's no pathogen present. It thinks one or more of the cells of your body are a pathogen and just destroys them. Diabetes type 1 is a perfect example of autoimmunity. Your body, if you're type 1, has destroyed the cells that produce insulin in the pancreas. So that is a type of autoimmunity. All right, helper T cells have receptors called CD4 receptors. Helper T cells are responsible for being able to activate the B cells to make the clonal expansion, make the army, right? And then also helper T cells are required for the cytotoxic T cells to make an army as well. And so that means that all of our memory, all of our lifelong protection depends on activation of a helper T cell, right? The helper T cell has to give permission to the B cell and the T cell to be able to proliferate and make those memory cells. All right. So 60 to 80% of all of your T cells are going to be helper T cells. And that makes sense, right? They have very important jobs to do. All T cells have to be activated by an antigen-presenting cell. And then activated helper T cells will secrete cytokines that give permission or turn on the pathway for the antigen-presenting B cell to proliferate, or for the cytotoxic T cell to proliferate. Okay, then we have naive T cells. They're matured, they've been released from the thymus, but they are not activated yet. And then we have cytotoxic T cells, which are the killing ones, right? So these have a different receptor called CD8 that they use to check the self tags. And any cell with a weird self tag is going to be targeted and killed. Okay. So here is a simplified view of this process. So here you have a macrophage. The macrophage is come into contact with a foreign particle and it's ingested it. It ID'd it as an antigen. And so then it did its job to display that antigen on its self tag. That tag is MHC class 2 because this is a white blood cell. Helper T cell has come to check the MHC2 tag. It uses a CD4 receptor to make sure that it's actually talking to a white blood cell. And it also has another receptor that will read the foreign antigen being displayed on the MHC2. So here, I know we didn't actually talk about two different receptors, but it is actually two receptors that are needed to read foreign antigen on the MHC2 tag. Okay, and then now this helper T cell, it's fully recognizing that it's antigens being presented to it. Okay, and so then it becomes activated. And if it finds any other cells, like B cells, that have also come in contact with that same antigen, it's going to say, yes, we have detected this antigen in the body. And it's going to secrete cytokines. You could draw a bunch of dots and write cytokines. And it's going to tell that B cell to go ahead and make that clonal expansion army. To proliferate means to make a bunch of copies of. And so you get that clonal expansion, and you'll have plasma cells that secrete antibody, and you'll have memory cells. Okay. So you cannot have great antibody response without T cells, right? If we didn't have the T cells, helper T cells, you would only ever get a small amount of antibodies and you would have no memory response. So T cells help B cells turn into antibody making machines and provide that lifelong immunity. T cells also can help fight, right? Cytotoxic T cells can fight directly. So in a bid to stop the spread of pathogen throughout the body, some T cells will trigger infected cells to self-destruct. And others will linger after the illness, right? Those memory cells, and they patrol tissues and make sure that that pathogen or germ cannot reestablish infection, right? So T cells are super important to our immune system. And this is one of reason why HIV is so devastating, is it takes out those CD4 cells. And so that really devastates the immune system. Okay, so what are CD8 cells? Again, are the cytotoxic T cells, sometimes abbreviated CTLs, cytotoxic T lymphocytes. Other times you might see abbreviated TC for cytotoxic T cell, or you might just see it referred to as CD8 cell. Unfortunately, all of those are used interchangeably. We see an abbreviation here, TH, that's a helper T cell. So CD8 cells need to be activated by an antigen-presenting cell, and then they need permission to proliferate by a helper T cell. Once this happens, then this guy here, the CTL, will tell the infected or weird cell to start its what we call apoptosis. So apoptosis tells the cell to cut up its genome, so it will no longer be able to divide and multiply. And while this cutting up of the genome occurs, it causes the cell to get in this very weird shape, and we call it blebbing. It looks like a blob, right? So this is blebbing. And in order for the apoptosis pathway to get turned on, this CTL, right, cytotoxic T cell, had to release perforin and granzymes. Perforin pokes some holes, allows the granzymes to get inside, and then turn on the apoptosis. And that's when you cut up the genome and bleb. So this bumpy weird guy is the cell that apoptosis got turned on. This nice looking round cell with all the little projections is the CD8 cell, the cytotoxic T cell. Okay. And so we see this image here. Now the process is being shown here. So this is the infected cell, and it's displaying on its self tag, on its MHC1, like that something has gone wrong, right? And so now a cytotoxic T cell comes around and reads that, yeah, something has gone wrong, and it releases the perforin, which makes the holes. That can directly kill the cell, but to make sure that we get rid of that, maybe the genome is weird, we don't know, whatever, we want to make sure that that cell is dead forever. And so we also have the granzymes that get in and start the apoptosis, and that will lead to the blebbing. The blebbing's not really shown here, like maybe the very start of blebbing is shown here, but you can draw that yourself. But cutting up the genome, you could write cut up genome and bleb, and that cell is dead, right? You've poked holes in it, you've cut up its genome, it's turned all weirdly shaped, it's dead. Okay. So in this case, the cell was infected by a virus, but it could have been a precancerous cell or cancerous cell, right? Or infected with the intracellular bacterial pathogen, right? Okay, so overall, we have a summary of the adaptive immune system. We have the humoral branch. Its job is to make antibodies. And we have the cell-mediated branch, and you can think of it as to activate cytotoxic lymphocytes. Make sure you read through this in detail, that you understand it, and that you could draw this from memory. Okay, so a little bit more about HIV. HIV is the virus, right? And a person can be infected with HIV and get treatment and never develop AIDS. So you do not have to develop AIDS if you get infected with HIV. Okay. And there's a lot of information in your textbook about HIV. We don't have time to go over it in all the depth that your book does, but I think it's very beneficial if you were to go ahead and read through that. So I put the pages here for the current version of the textbook, pages 547 through 554. There's tons of information about HIV on the CDC and the World Health Organization, WHO, as well. So there's no lack of resources for this. So in brief, remember, HIV stands for human immunodeficiency virus. It is a retrovirus. It starts out with RNA. It uses a special enzyme called reverse transcriptase to go from RNA to DNA. And then it uses another special enzyme called integrase to insert that viral DNA into the genome of the host. And in that case, that would be our T cells, right? You'll have a small amount of virus getting produced to infect more cells, but it's very low level. And because it's a low level, and because it then inserts its genome into the cell, it doesn't kill the cell. We really don't get we don't get symptoms usually. There's no specific unique symptoms for HIV. Some people never notice anything at all. Some people might notice cold or flu-like symptoms, but that's it. And so this is the dormant stage, right? Our latency. Okay. So it's evading the immune system. You're not getting a big immune response. You don't really know what's going on. And it can do this through several techniques. So the fact that it's integrating the DNA into the host cell, which is the latency, right? So that's one way. It can make special vacuoles. It can do special cell-to-cell fusion, and it can also change its antigens. And so all of these ways, these are all different mechanisms of escaping the immune system and infecting more and more of our T cells. If left untreated, it takes about 10 years, but you will eventually develop AIDS, which is acquired immunodeficiency syndrome. And so those CD4 cells, helper T cells, right? You normally have, 1,200 of these guys per mil. Another way to say milliliter is mm cubed, which you'll see a lot in the medical field. Anyways, so the T cell, the CD4 helper T cell count is going to drop to 200 or less. So it's such a significant drop, and that's when we start to see all the symptoms of AIDS. Now, it does really, it's the CD4 helper T cells that we focus on, but we will also see a decline in macrophage and dendritic cells. And perhaps that has to do with the job of the helper T cell. I don't know how much they're actually directly interacting with the macrophage and dendritic cells. Maybe someone will look into that. Okay, transmission is actually primarily heterosexual intercourse, right? We in the United States tend to really think of HIV as being a homosexual disease, primarily men. And really, HIV and AIDS is most prevalent in Africa, where we believe it's originated, and that was almost entirely spread through heterosexual intercourse. Also, pregnancy and breastfeeding will pass it on to children. Okay. Contaminated needle sticks can also spread it, as well as if blood transfusion was given and it was contaminated. Of course, we check for that in the United States, but not every country has the resources to do that. Okay, so it's an enveloped virus, as you recall. It will fuse to the host cell, releasing its viral RNA. So the unpacking stage, and then it will start the retrotranscription using that reverse transcriptase. Now you have this copy of double-stranded DNA, which we call cDNA. It's a copy. It's viral cDNA, and that is what's going to get inserted into the host. So now you have to go into the nucleus, and you need that special enzyme integrase, and it's going to plop it into the host genome. And it can stay there dormant, latent, when this cell divides, it will then pass on a copy of this to the new cell that it makes. On the same hand, a small amount of virus can be produced, so you can get the synthesis and assembly of a few new virus particles that can gently release and then find another cell. And so the process repeats. So latency is when the viral DNA hides in the host genome. And so phase 1, as I mentioned to you, there's no symptoms. It's asymptomatic. People only know they're HIV positive when they receive a screening, a test, right? So regular testing is important to catch it early. So if you diagnose HIV, it would be by looking for the presence of antibodies. So people will have to have been exposed 10 days prior. So if you have some event, you think that you may have been infected, you need to wait 10 days to get that test. But then you should follow up with another test 6 months later, just to make sure it's still negative. But anyways, if you test immediately, your body won't have had time to make any antibodies. We use ELISA, and if your ELISA test comes back positive, then we confirm with a Western blot to make sure. And then that's how someone would be diagnosed as HIV positive. And then they would receive multiple antivirals that target different parts of the cycle of replication, assembly, and release. And that will prevent HIV from replicating and destroying your T cells. And then you never develop AIDS, but you'll have to stay on that treatment, okay? If this does not occur, then the virus will continue to do its job, and you can enter phase 2, where you'll start to have infections that regular people don't have. So a lot of fungal infections, for example, or regular cold that will become very severe and turn into, bronchitis or pneumonia, right? So you're getting infections, you're more vulnerable, and then phase 3 is full-blown AIDS, where you basically don't have any immune system anymore, and you probably won't live very long, right? So it takes about 10 years to reach phase 3. Of course, at any of these phases, when someone discovers that they're HIV positive, they would receive treatment, and it's only during phase 1 that you can prevent entry into phase 3. If someone's already in phase 3 when they start the therapy, you can prolong their life. You're going to have to treat all of those opportunistic infections. Eventually, they will succumb to the opportunistic infections, will kill them. Okay, so this is why it's super important to have regular screening so that AIDS doesn't actually happen. All right, so a little bit more about antigens and antibodies. Now that we've completed our study of cell-mediated immunity. So I think this is a really good stopping point, and we will do the rest of the slides in part 2. All right, good job. Take a break.